So DNA replication is the process by which our cell copies its original DNA to produce two identical copies of that DNA. And one of the copies can be passed down to the offspring and the other copy can remain in that original cell. Now, DNA replication is known as a semi-conservative process. And this basically means that each one of the DNA copies that is produced contains one original strand and one synthesized strand. And to see what we mean by that concept, let's take a look at the following diagram. So this diagram basically summarizes the net result of DNA replication. So let's suppose we begin with the following double helix. Now, if the upper strand of DNA runs from the 3 to the 5 direction, then the bottom strand runs from the 5 to 3 direction because these two strands are known to be anti-parallel. They run parallel but in opposite directions. And these two individual single strands are held together by hydrogen bonds between the bases. Now, following DNA replication, we basically form two identical copies as shown. Now, notice that these black single strands of DNA are the original strands of DNA, and they are commonly known as parent strands or template strands, while the blue ones are the newly synthesized strands, and they are known as the daughter strand. So the process of DNA replication is semi-conservative because each one of these copies contains one of the single strands that came from the original double-stranded DNA, and the other one shown in blue is the newly synthesized strand. Now, how exactly do we initiate the process of DNA replication? Well, basically, a special type of enzyme, a special type of protein known as DNA helicase, moves very quickly along the double-stranded DNA molecule.